How's that going? Is the screen sharing relatively stable for everyone?
It is now.
That's good to hear. Yeah, let me just hide video panel. Okay, continue and present. All right, exciting stuff. Hello, everyone. I'm Kyle. Welcome to PSPlatypus, week 3. Let's get straight into it. Okay, so I'm going to start off by taking you through lipid metabolism. So basically, a really key concept to understand is the central role of acetyl-CoA. Acetyl-CoA stands for acetyl coenzyme A. It's basically an acetyl group, this blue part here, connected to coenzyme A, which sort of acts as a shuttle that transports the acetyl groups. And it's involved in so many biochemical pathways in metabolism of proteins, carbohydrates, and lipids. Its main role is in the Krebs or citric acid cycle, and that's to transport the acetyl group to be oxidized. And when it's oxidized, that produces energy. And it can also be used, though, to synthesize ketone bodies during starvation. And it can also be used to synthesize cholesterol for various functions in the cell. And just getting a bit more into the nitty gritty of the central role of acetyl-CoA. So yeah, it links together different energy sources. And I'm about to show you a diagram that will really capture that. For example, if you have excess energy, so you're taking in more energy than you need, than you're using, then you can store it as triglycerides, also known as TAGs, or that's the abbreviated form. And those can be stored in the adipose tissue and somewhat in the liver. And so, for instance, if you've eaten a lot of carbohydrates and you've got excess energy, then you can convert that into fat through acetyl-CoA. You can see carbohydrates that are broken down into acetyl-CoA and then reconstituted into triglycerides. And proteins, it's much the same. Sort of, you need acetyl-CoA as sort of an intermediary between the conversion of many, many different sources of energy. So you can see you do not need to memorize this diagram. This is a beautiful diagram put together by Sarah Damawan, I think of our year. Basically it shows a bunch of energy pathways. But you can see acetyl-CoA here in white, linking it all together. It really is playing a central role in metabolism of carbohydrates, lipids, proteins and amino acids, and even ethanol and ketone bodies. Okay, now on to lipoproteins. So they're sort of the key proteins that are involved in lipid metabolism. So they transport hydrophobic lipids in water-based fluids. So as you have hopefully previously learned, basically you have like dissolves like. So things that are hydrophobic will dissolve nicely in things that are hydrophobic. So oils will dissolve in other oils, for instance. Lipids will dissolve in lipids. And hydrophilic substances like water will dissolve in other hydrophilic substances. So for instance, ions, which are charged, will dissolve nice in a hydrophilic substance because water is also charged. And so what lipoproteins do is they transport lipids, which are hydrophobic, in water-based fluids, for instance, blood. And you have 4 main types. So you have chylomicrons, very low density lipoproteins, low density lipoproteins, and high density lipoproteins. So they're classed based on their density. And you've got this table that sort of shows the key ideas of them. But yeah, chylomicrons you use to transport dietary lipids. So just after you've eaten, your enterocytes, your intestinal cells, will make chylomicrons that are then transported to the blood, transporting those fats. So the way you can sort of see, I'll move to the next slide, but you can refer to this table for reference. So we're missing the VLDL here in this table, but this table gives a good visual and good idea of the makeup of different lipoproteins. So I've written here percentages are not accurate. I think they actually might be, so I'll revise that once we're done. but because that's talking about the specific percentage of lipids and percentage of protein. But here we go. So chylomicrons, you use those in dietary pathways. And you can see those mostly carry triglycerides, these TAGs, the yellow circles in this diagram. Very low density lipoproteins, not pictured here, come between chylomicrons and low density lipoproteins in terms of density. And they are involved in transporting lipids from the liver to body tissues between meals. So as you can see here, between meals. Low density lipoproteins, those transport cholesterol predominantly. As you can see, the cholesterol in blue, they're made up predominantly of cholesterol. And high density lipoproteins, they perform reverse cholesterol transport. So they take cholesterol from body tissues and return it to the liver, either for excretion or for use as for reuse in the body. Okay, don't get scared of all the diagrams. Don't get scared of the massive wall of steps. At the end of the day, it ends up being relatively straightforward. It's a pathway that just describes how fats that we eat get through to our body, how we break them down and how we distribute them to our body tissues. So I won't go through the steps in as much detail as we have here. But basically, you have dietary lipids. They get broken down and down and down and then they reach the small intestine. In the small intestine, they're taken up into the small intestinal cells, the enterocytes, and then they're built back into TAGs inside the intestinal cells. They're then packaged into those chylomicrons that we were talking about. And from there, those chylomicrons are sent out into the lymph, and then the lymph enters the blood eventually. And then so you've got capillaries here with the chylomicrons entering the capillaries. And then you have an enzyme called lipoprotein lipase. Now, lipase is something that breaks down fats, and it's related to these lipoproteins. So basically, it breaks down again these TAGs back down into their smaller components, free fatty acids and monoglycerides, to be used in tissues such as muscle for energy use and adipose for energy storage. Eventually, these chylomicrons, you can imagine the TAGs will have been used up, but the proteins won't have, right? These proteins, they're still just the package. And so you can imagine the sort of transport part of the lipoprotein, which is called a chylomicron remnant, it exits the bloodstream and it returns to the liver through these things called low-density lipoprotein receptors, basically the receptors for lipoproteins. Okay, then, and so that was the exogenous pathway. That's the pathway from outside. So when you're consuming new energy, it's entering your body from the outside and you're metabolizing that. The endogenous pathway has similar concepts, but it originates and ends at the liver, you can imagine. So it's endogenous because it's starting from the inside, from the liver, and then it's transporting these triacylglycerides and is distributing it to the tissues. So instead of chylomicrons, what you have is something called very low density lipoproteins, which are higher in density than chylomicrons, but lower in density than low density lipoproteins. So they're secreted, similarly, they're secreted into the bloodstream. They reach the capillaries. Lipoprotein lipase breaks down the triacylglycerides in them, which are then used as free fatty acids and monoglycerides in muscle and adipose for energy and storage respectively. Then they return, then basically they've been reduced in the amount of triacylglycerides, and triacylglycerides are less dense than a lot of other stuff. So when you lose triacylglycerides, you actually increase your density. So you become an intermediate density lipoprotein, similar to the chylomicron remnant for the exogenous pathway. And then those intermediate density lipoproteins can then sort of accumulate a bit more cholesterol, and then they can either turn into low-density lipoproteins, which is delivered to peripheral tissues, or it can return through low-density lipoprotein or intermediate-density lipoprotein back to the liver again for reuse, for replenishment. Okay, and the third pathway, once again, don't get scared by the diagram. So we have reverse cholesterol transport. So as I was saying earlier, you have the low-density lipoproteins, which transport cholesterol to tissues outside of the liver, extrahepatic tissues. So you can see we've got this low-density lipoprotein with cholesterol that's delivering cholesterol to extrahepatic tissues. But what if we have too much cholesterol? Well, then we do something called reverse cholesterol transport. And that's where high-density lipoprotein comes in. High-density lipoprotein, as we could see in this table, is predominantly protein. It's mostly made up of, see this red protein. And its job is to return cholesterol to the liver. So high-density lipoprotein precursors visit the extrahepatic tissues. They collect cholesterol, and then they return to the liver with cholesterol. Okay, so those were the three main pathways. I hope that was relatively understandable. I know I went through it very quick, but you can always look through the steps. And the more you go over it, the more straightforward it feels. It's really this sort of breaking down and reconstituting of triglycerides, and leaving the liver, returning to the liver. It ends up feeling a bit easier. And yeah, as James has said, feel free to chuck any questions in chat. I'm sure we have a few other PSPlatypus members happy to answer in chat. Okay, so now, well, how actually do we get energy from free fatty acids? Well, funnily enough, we convert them into acetyl-CoA. And the way we do that is through something called beta oxidation. So beta oxidation occurs in the mitochondrial matrix. And basically, each repetition of beta oxidation, it cleaves 2 carbons off of the fatty acid chain. So usually we have 16 carbon fatty acid chains. And so for a 16 carbon free fatty acid, you cleave it 7 times, 1, 2, 3, 4, 5, 6, 7, each time producing a 2 carbon long chain that you can load onto a coenzyme A to make acetyl-CoA. And it also produces other electron transporting molecules called NADH and FADH2. So for each cleaving, produces 1 NADH and 1 FADH2. So for a 16 carbon free fatty acid, you cleave it 7 times, you get 8 acetyl-CoA, because 16 cut into 8. It's 16 cut in parts of 2, you'll get 8 acetyl-CoA, and you'll also get 7 NADH and 7 FADH2. And then, of course, the acetyl-CoA can enter the citric acid cycle to produce energy, or it can be used for ketone formation or cholesterol synthesis. And finally, it's time to talk about ketone bodies. So ketone bodies, you produce them during starvation, and they're made from acetyl-CoA for use in glycolysis. So for instance, in a situation where you have low blood sugar, tissues will still need an energy source. And what you can do is you can get that acetyl-CoA from lipids and from proteins and convert it into, acetyl-CoA. And then that acetyl-CoA can be converted into ketone bodies. For instance, the brain only uses sugar and ketone bodies for energy. So if you don't have energy, if you don't have energy and you don't have sugar, you're going to need to use ketones, produce ketones and use them so that the brain is still getting some energy. And you have 3 different molecules that are ketones. You have acetone, acetoacetate, and beta hydroxybutyrate. And you can see in this diagram, don't get too intimidated by it once again. But what you've got is fatty acids. They're being converted into acetyl-CoA through that beta oxidation that we just talked about. And then from there, they're being converted into ketone bodies in a liver cell, a hepatocyte. And so they're being converted into ketone bodies, acetoacetate, beta-hydroxybutyrate, and acetone. And then acetoacetate and D-beta-hydroxybutyrate are exported as energy for the heart, skeletal muscle, kidney, and brain. Yeah, there you are. Okay, now on to the Great Metabolic Race.
Yes, oh yes, the Great Metabolic Race. I'll be taking that, and it's a quick couple of slides. I'll just share my screen real quick. Wait a second. Okay. Do you guys see it?
Looks good to me.
Okay, excellent. So Great Metabolic Race is essentially, we're just going to run through a scenario of a person running a race. And we want to talk about the changes in metabolism that occurs in that race. So this is the basic summary of everything you need to know. It looks like a lot, but I'll just walk you guys through it. I'll tell you guys where to look to make it a little bit easier. So in that table, you're going to see a time column and a primary energy source column. So he's going to start running, and before commencing the run, the primary energy source that's going to be used is going to be lipids undergoing beta oxidation, because beta oxidation is a process that we have for lipids to be converted into usable energy, like subunits such as NADH. In the bottom right, you'll see a graph, and I think that illustrates it pretty well. So typically at rest, you're gonna have fats contributing the majority of the energy because it's a high density energy source, and it's relatively, it doesn't, at rest, you don't need energy to be produced very instantaneously and very quickly. So it's only 30 seconds into the run that we use higher, we use energy sources that occur or provide your body with energy more instantaneously. And 30 seconds into the run, we use reserve ATP that exists in the muscle cells, and also creatine phosphate, which is another group of, it's another type of energy that's stored, especially for short bursts. And we undergo anaerobic glycolysis in the muscles. So anaerobic glycolysis, if you guys think a little bit back to your metabolism like chart, your diagram. So what happens is you have the glycogen, it turns into glucose 6-phosphate, turns into pyruvate. And in this process of glycolysis without the use of oxygen, you already have ATP and NADH produced. The reason why we undergo anaerobic glycolysis first is because 30 seconds into the run, our lungs haven't matched the the oxygen demand necessary to allow for aerobic glycolysis, which happens, like 5 minutes, which is right after. So what we have is a loop. So we're gonna have pyruvate being formed from glycogen producing NADH, and our pyruvate is gonna form lactate, which is gonna produce NAD+. And in this two-step process, we're gonna have an accumulation of lactate, but the balance of NADH and NAD+ is going to be relatively the same, since we have both NADH produced and NAD+ produced. So we're not going to have an excess of either, which means that we're not going to have problems in our electron transport chain later down the track. So since lactic acid buildup, you guys might know that it's related to cramps and stuff. I think lactate in and of itself can also be used as an energy source, and it undergoes gluconeogenesis, so we convert it back to glucose later. Not in this specific 30 seconds, but I imagine that happens later. So 5 minutes into the run, we're gonna have enough oxygen to allow for aerobic metabolism of carbohydrates. So if you have a look at the diagram on the top right, which essentially summarizes what's going on. So we have ATP immediately being used, creatine phosphate being used, and then anaerobic metabolism builds up a couple of seconds and a couple of minutes into it. And Uh, about 5 minutes into the run, aerobic metabolism begins, which rises at a steeper angle because of the amount of ATP yield that process has. So just a couple of minutes later, you're going to have aerobic metabolism provide the vast majority of your energy needs. 30 minutes into the run, if you have a look at the diagram on the bottom right, you'll see that eventually your carbohydrate, the energy produced by carbohydrate is going to slowly start dropping, and the amount of energy used by fats is going to start increasing because we essentially want to start using fats again. So lipids stored in adipocytes are mobilized and undergo beta oxidation, and in a 1-to-1 ratio, 30 minutes in. And since this is prolonged activity, we want to use the most efficient source of energy, which is going to be lipids. And at this point, it's not going to be the fastest source of energy, but that's fine. So we switch back to lipid oxidation being the primary source of energy into a 45-minute run, which is like a long run. I hope that was clear. If you guys have any questions, please chuck it in chat or ask now. But that would be the end of the Great Metabolic Race.
All right, lovely. I'll take the screen share back.
Yep.
Is that all right? Yep. Thanks, Deepak. Awesome. 1 second. Okay, now it's time to talk about ethics. So ethics are very important. They basically provide us a framework for moral reasoning. So ethics, yeah, they couldn't be more important. If they tried. I don't know how to overstate it. You need ethics as a way to reason about how you're doing good for the patient and for the wider community, which is, as you may be able to imagine, extremely important in medicine, where, you know, the perhaps not best, but perhaps most ubiquitous answer to why medicine is, to help people. We want to make sure that we are helping people and we're doing it in the right way. So you have 4 pillars of biomedical ethics, which are autonomy, non-maleficence, beneficence, and justice. Each pillar is important, arguably equally important, and they must be weighed against one another when making decisions. And the term meta-ethics is concerned with morality itself. So normative ethics, or what we generally mean when we say ethics, is talking about what we think about specific actions or decisions, or how to make moral decisions. Whereas meta-ethics is concerned with, well, what actually is morality? Where does it come from? Etc. We're mostly concerned with normative ethics. Okay, so for the first pillar of biomedical ethics, autonomy, it is the idea of respecting the patient's rights. So their own bodily free will, their ability to make decisions regarding their own body. It means that the patient has a final say, has the final say in their decisions about treating their own body. This is assuming that they are competent or have capacity to make a decision, referring back to Raymond's discussion of consent in med law last week. And there are 2 sides to autonomy. We both have a negative obligation to not restrict autonomy, to not impose restrictions that prevent people from being able to make free choices about their body. And we also have a positive obligation, which is to facilitate their ability to be autonomous in their decision-making, to be able to make decisions on their own terms. And I can see the final slide isn't completely finished, but basically autonomy requires that the patient be fully informed, and also that they not be under any sort of coercive effect. So it returns back to that idea of consent being voluntary, informed, and the patient having capacity. So it's autonomy to have autonomy, the patient has to be fully informed, and they also have to be able to make voluntary decisions and not be under some outside influence. That's what I meant to put here. Okay, so the second pillar is non-maleficence. It's that classic idea from the Hippocratic Oath of primum non nocere, first do no harm. Sorry if I butchered my Latin, I didn't do Latin. So clinically, as medical intervention will most often come at some cost or harm, it's really the avoidance of preventable harm. So, you know, when you stick someone with a needle, when you're giving them a vaccination, right, you are arguably harming them by puncturing their skin, right? And things like that. But it's for a greater good. And that discomfort of the needle is much less than the harm that would come to the patient through inaction if you were to not vaccinate them and they were to get a disease that could cause them much more harm. And you can see, though, that you have different levels of harm that could be preventable or unpreventable. So scarring and life-altering adverse effects. It returns back to that autonomy, the patient being fully informed. But at the base of it, it is fundamentally wrong to cause harm. And this harm could be caused either intentionally through malice, but it could also be caused through negligence. So not doing what you should be doing. Okay. Beneficence, similar thing to non-maleficence, but not the same. Non-maleficence refers to not harming the patient, whereas beneficence refers to doing good for the patient. It's really the point of what we do in medicine. And of course, you need to weigh the risks, the potential harms to the benefits of a procedure for the patient. And you need to enable the patient to do that for themselves as well, to weigh those, to make an autonomous decision. And finally, justice. So that's generally doing good for the wider community. In medical ethics, this often boils down to distributive justice, so the justice of distribution of resources. So if we don't have unlimited resources, then we need to make sure that those resources are being distributed based on need and with no consideration of the social characteristics of a patient. So as in HKS, equality versus equity is relevant. So each person should receive what they need, that is equity. If you give everyone a Band-Aid, but only one person actually needs a Band-Aid, right? You're using up a bunch of Band-Aids, and then what if more people come and need Band-Aids later and you don't have enough? You've given out a bunch of Band-Aids that you didn't need to. So this is why you need to be specific to the patient's need and not at all regarding their social characteristics, because that would be discrimination. Okay, now we have the concept of paternalism, definitely not one of the 4 pillars of biomedical ethics. It's another concept that really is very relevant in ethics and in ethical decision-making. So paternalism is this doctor knows best thinking. It's the overriding of autonomy when the doctor believes it is in the patient's best interests. Now, there are generally 2 concepts related to paternalism, and those are weak and strong forms of paternalism. So weak paternalism is generally good. So it's a doctor being able to help guide decision-making for a patient who doesn't have capacity to make an informed decision. So you can see why that would be good, right? A patient doesn't have the ability to make an informed decision. So the doctor is taking into account what the patient probably would want, what the patient's values are, and using that to help guide decision-making. Of course, definitely not coercing or anything. It's focused entirely on doing something for the good of the patient. However, a lot of paternalism, a lot of the discussion of paternalism will be around strong paternalism, which is bad. It's the idea of a doctor overriding a competent patient, so a patient who's able to make decisions, and overriding a patient who has made a voluntary or informed decision. So that's going, oh, well, the patient actually doesn't know this, they haven't considered that, in which case the doctor should probably be informing the patient rather than making a decision for them. Okay, confidentiality, another really important concept. And so it refers to the keeping of privileged sensitive information. So a patient has control of, should have, and does have control of their own medical information. They have a right to that because it is their personal information and it is very sensitive. And so confidentiality is the keeping of it in a secure, the keeping of this information in a secure way. It's the idea that as you'll probably learn to give in your OSCEs, the preamble about confidentiality for a patient, everything that we discuss here today will be kept confidential between you, me, and the treating team, unless otherwise required by law. It really creates a safe environment for the patient so that they can feel free to discuss things that might be a bit touchy, that they wouldn't want everyone to know. But it enables them to trust their doctor. And that, of course, would result in you actually being able to better understand what the patient is complaining of, what their concerns are. So there are some exceptions to confidentiality, sorry. And those most obviously are when the patient gives you permission to share their personal medical information. Well, then, yeah, you're able to break that confidentiality because they've given you permission, they've given you their consent. The other exceptions are to prevent harm to themselves or another individual. So if they say, for instance, that they're considering hurting someone, then you are required by law to disclose that to relevant bodies to make sure that people don't get hurt. Okay, and finally, let's just talk about the three main types of ethics, normative ethics. So they have three different focuses. So consequentialism is focused on the outcome. Rights-based theory, or deontology, is focused on rules. And virtue ethics is focused on moral character. So consequentialism is focused on the ends, the results. So a decision, whether it's moral or not, is based on the outcome. So this is sort of the ends justify the means thinking. And utilitarianism is an example of consequentialism. And it specifically aims for the greatest happiness of the greatest number of people. That's what utilitarianism is in layman's terms. Rights-based theory is focused on the idea that we can reach morality through rules. So by recognizing and upholding rights and other rules and ideals, we can help approach the idea that everyone is being having good outcomes. And virtue ethics is focused on moral character. So the idea of approaching morality from the idea of virtues. Virtues are positive characteristics. And the idea is that someone who is wholly virtuous will make the most moral decision in a situation. So you have many characteristics, but some of them are here, for example, that are virtuous, mercy, honesty, fairness, and integrity. But I'm sure you can come up with more on your own of what you think would be virtuous characteristics. And that sums up ethics. Thank you very much.
Awesome. Any questions for Kyle? Feel free, by the way, even if you have a question later on about anything else we've done today or in previous weeks, feel free to chuck it in the chat and one of us will get to it at some point. Easy peasy.
Thank you.
So I'll go on and share my screen. Give me a moment. Okie dokie. Hopefully people can see that. Yes, no, maybe. All right, we're going to assume that works. Full screen. All right. So health determinants is basically when we look at the deeper causes of disease, because we know, like, of course, you can go up to a patient and say, well, to use the example in the first row there, this patient has stomach ulcers. there's a chemical imbalance in his body, which is causing him to have lesions in his stomach. And that's what's causing his stomach irritation. Awesome. But we also want to know why he has that chemical imbalance in the first place. And so we need to go up and up and up and up. We want to go back along the chain of causes to the start. We want to try and get the most base causes of health outcomes, of diseases, because those can be the most effective in actually treating health. We remember to learn about the biopsychosocial model of health, in which we really have a more comprehensive view of what health is. And as part of that, we also need to recognize that health problems are not only caused purely by physical issues, but also by deeper social and societal issues. So if those can impact on health, we need to be aware of them and we need to be able to deal with them, basically. So we talk a lot about like upstream and downstream issues. So the upstream causes will be the initial things. So these are things that happen on a bigger picture, or they can happen to a single person, but they're not strictly health related, or at least we need to dig a little deeper to see how they're impacting on health. So something like unemployment. How does unemployment lead to stomach ulcers? So if you're unemployed, you're probably stressed. If you're stressed, you get things like chronic inflammation, because your body in a state of stress starts to release inflammatory hormones, basically. And if those are pervading your entire body, then parts of your body which shouldn't be inflamed are going to be inflamed, and then you get stomach ulcers. So we've gone from an upstream cause, which at first glance seems like a very indirect cause of disease, and we have now seen how it can very directly cause disease in a person. And that's why we need to care about these things, because they are relevant. And of course, more big picture stuff like displacement due to conflict. If you have people like refugees, if you have asylum seekers, if you have internally displaced people where their homes have been destroyed or something, but they're in the same country. These people, they often have malnutrition because they're not getting food. They often have poor hygiene. They have several parasites and untreated infections because they don't have access to either good food or good medical care. And of course, those are going to result in worse health outcomes. There's more at play here than purely the physical. And of course, health legislation is another realm in which we can have impacts on health. Because if you have poor environmental legislation, for example, you might let carcinogenic chemicals into the environment, and then you find out a few years later, your population has a spike in cancer rates. So that is another area where a decision which was not immediately health related had very clear and defined, or even unclear, but still important health effects. All right, so we talk about the structural determinants of health, and these are the really big, the sort of the highest level classification. So on a geopolitical level, we have things like conflict, displacement. So if there's a war, if there's a widespread natural disaster which causes people to be displaced, their homes are being destroyed. And we can have, to a degree, globalization, which can have various impacts on society and can also lead to things like extractive institutions where you empower corporations and companies to, for example, go into third world countries and take advantage of mineral wealth and that sort of thing, while not distributing that to the populace, or in other words, exploitation of these countries. So even though that seems at first glance to be a very political issue, we can see how it impacts on health, because if there's less funding going to these countries, then there's going to be less money to buy drugs. There's going to be less ability to access good healthcare. And those are all important to health. On a socio-cultural level, so this is zooming in a little bit, we can have things like cultural blindness. This is a concept you'll be talking a little bit about, especially in relation to Indigenous people. And it's basically the idea that we need to be aware of cultural differences. And when we're not aware of those cultural differences, we can have very direct impacts on the ability of people to feel safe, the ability of people to feel trusting of medical institutions and medical practitioners. We've also got the social determinants of health, which we'll be looking at in more detail in just a moment. Now, on an environmental level, the environment we're in contributes a huge amount to our health. So like, obviously there's very clear stuff. Like if you live in a city with a lot of smog, your respiratory health is going to suffer because there's lots of particles there and your body's not equipped to deal with those. At the same time, we can also have issues where people have inadequate space to exercise, things like that. We can also have, like I said before, carcinogens. We can have lots of things in the environment which impact on people. And we can also have historical and legal factors. So we have in Australia and several other post-colonial nations, we have sort of lingering impacts of things like racial discrimination against Indigenous people. We have all sorts of big health inequities which came out as a result of history. And we also have, in the purely legal sphere, we have current legislation and health policies. So what are the government's policies relating to both how they administer the health system and also how do they protect the environment, how do they encourage healthy living, how do they all do all these things? Those are quite important. And demographic shifts are more of a historical factor. So this is talking about, say, if we have an aging population, that presents health challenges. How are we going to deal with having more and more aging people with more and more different health issues? So that's another thing which can impact on health on a societal level. Now, the other thing that's important to understand is it's not like, one of these is going to be relevant. You'll most likely have multiple of these relevant in any given case. So say you have a refugee from Somalia who's come to Australia. So on a geopolitical level, there's been conflict. They've been displaced from their home and they've had to come to another country. We can have, to a degree, a legal impact. So say if they're on a refugee status, which doesn't allow them to access Medicare, then they might be disincentivized to seek healthcare. So that's that's a legal impact. And then on a socio-cultural level, if the doctor doesn't understand their culture and makes them feel unsafe in a consultation, then they may not be going back. And so that's a failure of the health system to give them good access to healthcare. So you can see how a real world case where we actually look at this in a real world context is going to have more than one of these factors. And so it's important to understand that they can interact with each other and actually lead to even more serious impacts. All right, so this is sort of the main list of what we call the social determinants of health. There's a little bit more on the next slide. But these are basically zooming in on social issues. So this is the sort of thing that you get within many, not necessarily only Australia. You get these within every country, but it's the sort of thing you could imagine happening to someone within Melbourne. Some of the bigger picture geopolitical stuff can be a bit harder to sort of understand in a local context, but all of these can definitely happen right where we are right now. So some of these are more obvious than others. So things like unemployment, of course, if you have less money, if you have less stable employment, then it's going to be harder for you to use health services. And you might say, okay, well, maybe I don't really need to go to the doctor. It's a bit expensive. I don't know if I'm going to have enough money. I'll just, leave it. It'll probably get better, that sort of thing. And as we talked about before, stress can actually have very direct changes to your body's function because it really does a lot of bad things to your body to have chronic elevated stress. A little bit of stress is okay. A lot of stress is not good. And we can have developmental trauma. So if you have interrupted development in your early childhood, then that can have a serious impact on your later life. And really decades down the line, we can have impacts of early developmental trauma. Social exclusion. So this can be any number of different things, from racism to simply feelings of isolation. And these can have, again, direct and indirect effects. So your psychological health, remember, we're thinking about health not only as biological, but also psychological. Your psychological health is going to be affected immediately. And you can also have, again, long-term stress, elevation, and that can lead to physiological changes, which will eventually turn up at your door as a doctor. Transport options is a thing that is a bit sort of obscure. You think, okay, well, that is kind of related to health, I guess, if people aren't walking, but it's actually more than that. It's not only the ability to like how sedentary your lifestyle is, but it's also about, are people able to access jobs to get them money to keep them out of unemployment? Are people able to access healthcare? Are people able to access education? So these are all quite important factors. And again, you can already see how they interact with each other. They don't exist in a vacuum. They exist with one another. One of these can impact the other, one of these can bring about another one. All right, we've also got access to healthy food. That one is pretty self-explanatory. That's one of the most straightforward of this group. We can also have religion, which is an interesting one, because although most of these are sort of purely negative impacts, religion can actually have a positive or a negative impact. So we have research which shows that there are positive impacts on health that are associated with certain people practicing religion. But at the same time, it's not guaranteed. We can also have social exclusion and things like that. So practice of religion can actually lead to negative health impacts. So it's really situational. And all of these are going to be different depending on what the particular patient situation is. So caste and discrimination, this is sort of a proxy for both discrimination on things like race and also of things like class. So this is healthcare, education, and jobs. There may be legal or social barriers or institutional barriers to people seeking better healthcare, better education, better jobs. And again, we can have more interaction with different factors here. So social support is probably the only purely positive one of these. So if you have social support, we have sort of flow-on effects. And that can not only be just because you're feeling better psychologically, it can also be because people with a better social support system, they're more likely to go and seek help from a doctor. They're more likely to feel comfortable accessing healthcare. And addiction, of course, that's a very insidious one. If you're addicted to a substance, then you're going to have physiological impacts on your body. If you not only if you keep using it, but also if you stop using it and you go through withdrawal, but also psychological impacts on you. So there are a couple other concepts we want to understand here. So marginalization is sort of the concept of a certain group being ignored, basically, or deprioritized by governments or other institutions. So like I said before, in Australia in particular, the reason we care a lot about Indigenous health is because we have a very long history of discrimination, literal genocide in the early stages of colonialism, and political marginalization, which to a degree continues to this day. And so we have really, really big inequalities and inequities between Indigenous groups and the rest of Australia. And you can see those even now in statistics, which you'll see when you do Aboriginal health. But these are really big and long-standing inequities. And so there can be a very huge impact over the long term on the health of an entire group of people because of marginalization. And we also have the concept of structural violence. So, the classic idea of violence is a very physical one, you hit someone or something. Whereas structural violence is talking about how injury to individuals can not only be a case of simple physical thing or verbal things, but it can also be because of the way institutions are organized. If we have exclusive institutions, if certain people are shut out of the socio-political framework, we can have injury both on a psychological level and to a degree on a physical level, because if people are not able to seek good healthcare, if people are cut off from social services, then they can have very direct health impacts. All right, that brings us up to the start of our genetics topics. I'll hand it open to Marwan, unless there are any questions first, which I'll try and answer.
Thank you, James. All right. So now we're going to talk about genomics and population genetics. And we start by the definition of genomics, which is the study of whole DNA of organisms and the elements that are associated with this. Part of genomics is personalized medicine or precision medicine, which is an approach to disease treatment and prevention using genetics, considering also individual variability in genes, environment, and lifestyle. So there are advantages and disadvantages for genetic testing. Their benefits are prescribing more effective drugs, avoiding drugs that cause significant side effects, as well as it focuses on prevention rather than treatment. Like it prevents the diseases from happening in the first place. The disadvantages of genetic testing, including it can cause unnecessary stress because the results of this genetic testing are not 100% correct. There are also privacy issues, and it can cause some possible discrimination as well as ethical problems associated with the autonomy of the patients. One of the applications of genomics are pharmacogenomics, which is a prediction of the individual response to certain drugs using genetic information. So this means like different individual responds differently to certain drugs. And why is that? This is because the variations in genes encoding drugs metabolizing enzymes in our bodies. So how drug acts in our body depends on our genes as well. And there is a variation between individuals. Another application is called the single nucleotide polymorphisms, or SNPs. So SNPs basically is a variation in the DNA sequence that occur in certain, in a population in a single with a single nucleotide differs between members of the species. We inherited like 3 billion bases from each of our parents, and the difference between these bases is they differ only by 1 base per 1,000 base, and this is like universal. So the genes of any 2 random people from anywhere, they differ by 1 base per 1,000 base. And the SNPs are the common differences between individuals. It means that these mutations are not rare. And that's why we need at least 1% of the population that have the same mutation to be considered as SNPs. SNPs can occur also in exons, introns, and intergenic sequences. And if we divide the mutations of SNPs, so 50% are in the non-coding regions. This could impact the could have an impact on the promoter activity or pre-mRNA conformation or stability. 25% can cause missense mutations, and the rest 25% can cause silent mutation. They are silent. So we have 2 projects, main projects regarding the human genome. The first one is the Human Genome Project. It lasted 15 years, and the aim was to determine the human genome so that we can underlie human diseases and diseases susceptibility. Another project, if we can call it a project, is the genome-wide association studies. So we have a very large sample size. We have lots of people who have this genetic variation, and these people who doesn't. So we can consider them as a control group. And we use SNPs in these studies. So these studies can find the differences between populations, and this helps to suspect and underlie diseases. Then we'll talk about population genetics. It's very important because it considers the prevalence of genetic traits in population, the frequencies of carriers, and the ways how traits are passed from one generation to another. This will enable us to calculate the probability of inheriting a genetic condition or genetic disease. And then we have the definition of the gene pool, which is the total number of all alleles in a population. So allele frequency is the percentage of that allele in a population. If we have an allele, so we will call it like A. We will name it A. The total number of all A alleles in a population is equal to the number of individuals that have the homozygous of this dominant allele multiplied by 2, plus the number of people in this population that have the heterozygous of that allele. The same with the recessive allele. So the total number of people that have this recessive allele is the number of people that are homozygous to this recessive allele multiplied by 2, plus the number of people that are heterozygous to this allele. Therefore, we can calculate the frequency of the dominant allele if we say like it's P. So P equals the total number of A alleles in a population divided by the gene pool of this population multiplied by 2, and the same with the recessive allele. Genotype frequency is the proportion of that genotype in that population. So there is a difference between genotype frequency and the allele frequency. With genotype frequency, we are focusing on the genotype. So we have 3 possible outcomes, whereas in allele frequency, we are just focusing on the allele. So for each trait, we have just 2 alleles. And the genotype frequency is the number with a particular genotype divided by the total number of individuals. And it actually equals like the genotype AA, the homozygous dominant trait is equal the allele frequency of the allele frequency of the dominant trait squared, and the genotype frequency of the heterozygous trait 2 multiplied by the allele frequency of the dominant and multiplied by the allele frequency of the recessive traits. And then the genotype frequency of the recessive traits equals Q squared, which is the Q is the allele frequency of the recessive allele. And then we have Hardy-Weinberg law, which states that in large randomly mating populations, allele frequencies do not change over time. What does that mean? It means in a very large populations, dominant traits do not undertake recessive traits completely. But here we have must have some conditions, like the population must be large, it must be randomly mates, so there is no migration, no selection, and no mutations. So we can say that this does not exist in real life because we can't control migration, we can't control genetic drift, we can't control mutations. But through Hardy-Weinberg law, we can calculate the expected genotype frequencies and then compare it with the actual genotype frequencies. Here we have the equation of Hardy-Weinberg. So it's P squared plus 2PQ plus Q squared equals 1. If we have P or Q, we can calculate the other one. So if the expected and the actual genotype frequencies are similar or are basically identical, we can say that this population is in Hardy-Weinberg equilibrium. There are some factors affecting the Hardy-Weinberg equilibrium. So mutations, because they bring new alleles, genetic variation, migration, which is the movement of gametes between populations. This also causes new alleles to be added to this population. Selection, because according to Hardy-Weinberg law, this must be randomly. So selection is not considered as random mating. Also genetic drift. We need a large population, and genetic drift will cause the population to decrease in size. Finally, non-random mating. And that's it. Thank you so much.
Thanks very much to Marwan. So we've got one more topic, chromosomal disorders, just to round off a little bit on genetics, basically. Feel free to drop any questions for Marwan in chat. Okie dokie, hopefully you all can see that. Right. So chromosomal disorders is basically talking about genetic variation, which occurs on the level of chromosomes rather than of individual genes. So it can impact individual genes, but we're talking about much bigger changes to your genetic structure than we were in the previous parts of this genetics area. And so we focus mostly on disorders, because when you have big changes like this, it usually results in big impacts on you. And a lot of these can be very bad for the patient. So unlike things like SNPs and certain mutations where you can have sort of an innocuous thing where it just changes something, but not necessarily causes disease, these very often cause disease. Okie dokie. So chromosomes are like a highest level of DNA organization. You have your DNA and it gets wrapped around histones, and then you have chromatin, which is the combination of those two together. And then it gets all wrapped up, and it makes the chromosome. So we can exist in a chromosome in two forms. So we can have a chromosome existing in two forms. So for most of the cell's life, it exists in the form of one chromatid. So it'll be just like you can see the diagram, there are two of them on the screen. So there's a smaller one, the green and purple one, and then the bigger black and white one. Those are both one chromatid. But we can also have this sort of classic X shape. So that's when you have two sister chromatids, and the sister chromatids would be joined at the centromere there. That's what it's for. But it appears like a bit of a cinched line because the centromere is a little bit thinner than the rest of the chromosome, basically. So we have a little bit of terminology here. We use P to talk about the short arm, P for petite, and we use Q for the long arm of the chromosome. And we call the little tip of each arm the telomere. So you're going to hear a lot about telomeres from Craig Hassett. I don't know if you guys had a HEP lecture yet, but he cares a lot about telomeres, and he finds a way to link them into almost everything. Telomeres are basically a bit of, for lack of a better term, junk DNA at the end of your chromosomes. So they don't code for anything super important. They're basically there because every time you copy your chromosomes, you lose a little bit off the end. So they're like a buffer. So we can lose this much material before we start to actually lose important genetic material. And so shorter telomeres are correlated with more genetic errors. Longer telomeres are sort of correlated with longevity. And that's sort of an area of ongoing research. But for you guys, what you need to remember is telomeres are on the end. We have P for the short arm, Q for the long arm. And we have something called G banding, which is when we stain a chromosome, they have light and dark bands. So it looks like there's a whole bunch of little stripes on them. Each chromosome will have its different banding pattern. So if I look at a chromosome 1 in somebody and a chromosome 22 in them, they'll have, first of all, very different sizes, and they'll also have different banding patterns. But if I look at a chromosome 22 in me and a chromosome 22 in a different person, it's probably going to be a pretty similar banding pattern. So we allow, so the banding pattern lets us identify which chromosome is which when we do something called a karyotype, which we'll talk about in a sec. So not only do we have the short arm and the long arm, but we also have what we call an ideogram, which is basically just a way of recording how far away from the centromere a gene is on the chromosome. So to use the little example there, I have 5q31.3. So I know this is on 5. So this is chromosome 5. We've got a little diagram there to the right. I know it's on Q, so that has to be the long arm. So I'm now looking down Q, 31.3. So I start at the centromere, I go down, down, down. The numbers will go higher as I go further from the centromere, and then finally I arrive at 31.3. And that's a little segment of DNA of a certain coloration, basically, which I know contains the gene that I'm looking for. So that's our way of like precisely determining where a gene is. All right, and so we can classify chromosomes based on where the centromere is, because it varies quite a lot between different chromosomes. So we call them metacentric when we have the chromosome when with the centromere more or less in the middle of the chromosome. So the short arm and the long arm are pretty similar in length. It's much harder to tell which is which. When we have a submetacentric chromosome, it's much easier to tell which one is which, because the short arm is very short, the long arm is very long, relatively. We can also have acrocentric chromosomes. So that's where the centromere is very close to the end. And then the short arms are what we call satellites. So they're very tiny little things, and they usually encode ribosomal RNA. There's another fourth type. You don't need to worry too much about this one. They don't really cover it much in the course. And that's basically when we have the centromere at the very end. So there's literally no short arm. But you don't need to worry about that too much. The first three are the important ones. Okie dokie. So the human karyotype is basically, we take a picture of a cell when the chromosomes are condensed, so during mitosis usually. And then we find all the chromosomes, we sort of cut them out of the image, and we order them all. So it's not like the chromosomes in your body like sort of nicely line up like this. They'll all be jumbled around in the cell, but they'll be condensed because they need to be pulled apart for mitosis. So we take a photo and then we sort of find each one using the banding pattern, and then we arrange them like this. So the classic order you can see here, it goes 1 to 22 because we have 22 pairs of autosomal chromosomes. So that's just ones that are more or less the same for everyone. And then we have 2 sex chromosomes. So of course, males have XY, females have XX. So we list the 22 pairs of autosomal chromosomes first, and then we list the 2 sex chromosomes afterwards. So a normal human cell will have 23 pairs, 22 autosomal pairs, 44 autosomal chromosomes, and 1 sex chromosome pair with 2 sex chromosomes. So we can also use a little bit of sort of notation when we talk about what's in a karyotype. So first thing we do is number of chromosomes. So for a normal karyotype, like the one on the right, human male, we have 46 chromosomes because we have the right number. Our sex chromosomes are XY because we're a male, and we can see, of course, the X and the Y chromosome, they're quite different. And then we have abnormalities. Now, this one doesn't have any abnormalities because this is a normal karyotype. But if we had an abnormal one, we might do different notation. And I'm going to give you example karyotypes for all of the disorders we're about to talk about. So there are a few different ways we can have a chromosomal abnormality. The first one is if we have a difference in the number of chromosomes. So probably the main category you'll be dealing with, because it's the cause of most of the actual issues, is something called aneuploidy. So we're missing a chromosome, or we have an additional chromosome. So monosomy is when we lose a chromosome. So mono 1, so we only have 1 chromosome when we should have 2. So we've lost a chromosome somewhere. So we could have, for example, a karyotype. We have 45 chromosomes because we've missed 1 out of 46. We have XY because this is a male in our example. And then we have minus 14 because we're missing the 14 or one of the 14 chromosomes. So most of the time, this is lethal because we're just losing too much genetic information. Now, exception to this is when we have females who lose one of their sex chromosomes. So usually a female would be 46XX. In this case, the female is 45X, so they're missing 1 sex chromosome. But because the X chromosomes have a bit of redundancy, think about it, your males only have 1 X chromosome, so you can survive on 1 X chromosome. But as we're about to explore, that does lead to certain disorders, like it's a disorder in of itself, so there's going to be impacts on the patient. Trisomy is when we have 3 chromosomes instead of 2. So an example karyotype, we have 47 XY, this is a male, and plus 21. So if you guys know about trisomy at all, you might be able to identify that as Down syndrome. And so we can often have very serious mortality impacts. So there are really, really extreme cases, levels of infant mortality among people with trisomy. But Down syndrome is actually an exception. It's actually well known because it's not as lethal. So it's the one where infants with trisomy 21, Down syndrome, they're going to have less, they have more chance of living, basically. We have tetrasomy, which is when you can gain 2 chromosomes. So you have 4 chromosomes. This is super rare. So we're not going to talk about any disorders using it because it's just too rare, basically. We can also have polyploidy. So this is when you have more sets of chromosomes than you're meant to. So you're meant to have 2 sets of chromosomes. So you have 23 and then you have 2. So there are like 46 total. That's what we call 2N. Your haploid number is 23 for a human. It's different for other creatures. But if we have 3N, for example, we're going to have 69 chromosomes total. So in our example here, this is a female, and they've somehow got a 3N cell. So they've got 3 sets of chromosomes and 3 sex chromosomes. So this is often lethal if this just happens in normal cells, but in a certain type of cell called a megakaryocyte, you might learn about this a little bit later. It's involved in platelet creation. This is normal. So they actually operate on increased haploid numbers. But if you just have this happening in normal cells, it's probably a big deal. All right, we can also have structural abnormalities. So this is basically, instead of just missing or gaining 1 chromosome, we have errors with we have errors with the chromosomes themselves. So within 1 chromosome, we can have deletion, which is pretty self-explanatory. We delete a section of the chromosome. We can have duplication, which is where 1 section of the chromosome erroneously gets copied twice. And so we have, it gets repeated in the new one. And we can have inversion, where it sort of flips over. Between our chromosomes, we can have translocation, which is basically like trading or moving genetic material. We'll talk about the specific types of that in a second. Or we can have insertion, which is basically just, it goes from 1 chromosome to another chromosome. So 1 gains information, 1 loses information. And so this happens when chromosomes break apart at some point, and then they rejoin in a weird way. So apart from just changing genetic information, we can lose or gain genetic material. So if we lose genetic material, then we might lose an important gene, and then you're in big trouble. We can also have a gain of genetic material, which can also have its own issues, even though we technically have all the genes we need and more. So in a balanced rearrangement, that's when neither of those happen. So we don't have a loss or gain. We have the same amount of genetic material. It's just in a different organization. So this is usually okay, but there are 2 issues with it. So the first one is when we cut things out and then move them, for example. So when you have inversion, for example, the bit flips over, the cut might come in the middle of a gene. And if that cut happens in the middle of a gene, then that gene is kaput. That gene doesn't work anymore. So if it's in a major gene, then that can actually be quite a big problem to you. And the other thing is that you can have unbalanced arrangements in offspring. So let me try and explain how that happens. So if I say I have my chromosome, let's say 14 on a whim. So one of my chromosome 14s undergoes a translocation. So it loses a bit of information, and the information goes over to the other chromosome 14. So for me, that's fine. I still have all the same genetic information. I'm okay. But if I then have a kid, then the kid only gets one of those chromosome 14s. So remember, one of the chromosome 14s is missing genetic material, and the other chromosome 14 has too much genetic material. And the chromosome 14 that the child will get from its other parent is going to be a normal chromosome 14. So in the end, that child will either have too much genetic material or will be missing certain genetic material. It's no longer a balanced rearrangement. It's now an unbalanced rearrangement because there's been a net loss or gain. Hopefully that's understandable. Right, so we've got a few different types of translocation. So reciprocal is basically the ends break off, and then instead of going back where they were supposed to be, they go to the other one. Insertional is where we cut a bit out, and then we put it into another chromosome. So it's the same thing. Information goes from one chromosome to another chromosome, but instead of at the ends, it's in the middle somewhere. So you might expect, this one is a bit rarer because it's harder for this to happen basically than reciprocal. And we also have something called Robertsonian. So this is when we have 2 very short chromosomes already. And then what they do is they actually just take the whole chromosome and stuff it on top of the other one. So overall, we lose one chromosome because we're meant to have 2 chromosomes and we now just have one big one. But we still have most of the genetic material. We lose a little bit of genetic material in those short arms. This is random fact, but this is actually thought to have occurred in certain parts of like genetic evolution of certain species. So obviously it doesn't always result in death, but it can obviously often lead to issues. Okay, so we have, like I said, a lot of trisomy. There's a lot of disorders which occur from trisomy. So Patau syndrome is trisomy 13, trisomy of the 13th chromosome. So your karyotype is going to look like 47, one more than 46. We used a male in this example, but it could well be a female. And then we have a plus 13 because we've got one more 13 than we need. So not a huge incidence, but if you think about it, 1 in 5,000 still means quite a decent amount of people being born with it. And this is one of the ones with very severe infant mortality issues. So the child has a very low chance of surviving. And even if they do survive, there's going to be pretty serious effects. Same with Edwards syndrome, so trisomy of the 18th chromosome. This one is a little bit more common. And there's quite severe issues and you're unlikely to survive. So Down syndrome, you probably know about, like I said, because more people with it live. So more of these people survive and then grow up. And of course, they have certain developmental effects, but it's not anywhere near on the same level as Patau and Edwards. So the incidence is more like 1 in 700, which is obviously several times more common. And if you have more people surviving, then in the general population, you're going to have a lot of people who actually grow up with Down syndrome compared to Patau or Edwards syndrome. So Cri-du-Chat syndrome is a little bit more obscure, so you probably won't be asked too much about it. This is where we have a part of chromosome 5, which is missing. And the reason that this one in particular is distinctive is because the infant's cry, so when like the infant is crying, it sounds like a cat. Don't ask me, I didn't name it. All right, so we have a few sex-linked disorders. So basically, these are ones that affect the sex chromosomes, whereas the previous ones only affected the autosomal chromosomes, the other ones. So Klinefelter syndrome is when we have a male with an added X chromosome. And this one like often goes under the radar, so people might not realize they have Klinefelter syndrome until quite late. So they can have certain feminine features, so like additional breast mass, for example. They can have infertility, and they can have learning difficulties. So I'm sure you can notice, like if, for example, your major symptom was infertility, that's going to be pretty hard to notice until you, for example, start trying for a baby. So that can go under the radar for quite a long time before you actually realize, hey, I've got Klinefelter syndrome. When we do have it, we treat it with testosterone, because one of the big reasons we have all those symptoms is because the added X chromosome leads to a hormone imbalance. Turner syndrome is the one, this is actually the one I talked about when I talked about how monosomy has one exception to like livability, which is Turner syndrome. We have one X chromosome, the patient comes out female, but has undeveloped secondary sexual characteristics. So the primary sexual characteristics are the genitals. The second ones are things like breast and pubic hair. And they have, sorry, that should say redundant neck skin. So like skin folds at the neck, and short stature. So it's treated with growth hormone for the short stature and estrogen as well. Okie dokie. Sorry if that was a little bit fast. Let me know if there's anything you'd like me to clarify. But that pretty much brings us to the end. We have a couple of questions in chat. All right, looks like a few have been answered. Okay, let me just go over these real quick. One week the chromosomes have light and dark bands when stained from Kellen Munns. Okay, but sorry, Marwan's already answered that. Will's answered the one about centromeres. If a chromosome is metacentric, how do we tell which one is P and Q? Basically, people before us have already assigned one P and Q, and because of that banding we talked about, we know which one corresponds to the P and which one corresponds to the Q. So it can get a little bit arbitrary, but like we know this end is P and a certain end is Q. So when I say it's how long the arm is, it's not like we look at a chromosome and if a certain arm is longer, we call that one the Q arm. It's rather that in 99.99% of people, this is the way the chromosome is displayed. And so we call this one the P and the other one the Q. So it's based more about like the general idea. Extra information. Look, so the characteristics I meant, sorry, that was the, I don't know 100% what you were referring to there. Abdullah, how much detail do we recommend memorizing metabolic pathways? You don't really need to memorize them. It's more, okay, how do I say? A lot of them go into a lot more detail than you need. So like glycolysis, for example, it has like 12 steps. You don't need to memorize 12 steps of glycolysis. Like that is ridiculous. So there's certain ones that are important to know. The lectures will do a good job of showing you that. And we've shown you this. We haven't really been talking about metabolism past the initial lipid metabolism stage here. But we've shown you like how the different stages go. And that's more important than particular like reactions. So like the fact that you have this precession from chylomicron to chylomicron remnant, VLDL to IDL, that sort of thing. Why can you not see the extra on the karyotype? So like I said, chromosomes can either be one chromatid or two sister chromatids. So if it's a karyotype of the single chromatid, then it's only going to be a cinched line. Should we memorize the types and numbers of inputs and outputs for metabolism? If it's like cellular respiration or something really basic like that, then that's probably useful. But once we get to more obscure stuff, it's not quite as useful. Like the lipid metabolism stuff, if you guys remember, like when we were converting to acetyl-CoA, it's good to be able to do the maths on that one because it can be relevant. But for a lot of the other ones, it's basically too obscure to be important. So we've gone into more detail on the places where you should be focusing. One more from Cody Chan, what causes the change in density of lipoprotein? Okay, so fats are not very dense, proteins are very dense. So at the start, we have a lot of fat, which are a lot of very like low density stuff. And then when we lose the low density stuff and it goes into the tissue, we get a more dense remnant. It's like we have like a little golf ball wrapped in cotton candy. If we take away the cotton candy, then the remaining ball will get denser and denser and denser because we're only leaving the dense stuff back. All right. Feel free to stick around if you've got any other questions. Other than that, I think we're more or less done. I'll see you guys next week. Have a good one.